Hi friends, this is Varsa Subbarao along with Horti Buddy welcomes you to Green Coach Horticulture. Green Coach Horticulture is an online tutorial for various horticulture subjects, and today we are dealing with the plant breeding. And today's topic is the importance of mutation breeding. Let's start our today's class without any delay. Mutation breeding. Mutation breeding. First, the mutations are discovered by the scientist Hugo de Barres. Mutations are created in plants by using X-rays by Stadler. Mutations in Drosophila are identified by Muller. Chemical mutations are first time used by Robson. What do you mean by mutation breeding? Using of induced mutations or artificial mutations for the crop improvement can be termed as mutation breeding. Hugo de Barres first time termed mutations, and mutations in plants are created by using X-rays by Stadler, and mutations in Drosophila are identified by Muller, and chemical mutations are used by Robson. Point mutations. Point mutations means these mutations occurs only in nuclear genes, means only one nucleotide changes. Point mutations means it occurs only in nuclear genes, only one nucleotide changes. What do you mean by gene? A bit of DNA that can code for a functional polypeptide chain can be termed as a gene. A bit of DNA that can code for a functional polypeptide chain can be termed as gene. Mutations. Mutations are of two types: spontaneous mutations, induced mutations. Spontaneous mutations, they occur spontaneously in nature, means we don't induce these mutations, they occur naturally. The frequency of spontaneous mutation is 10 to the power of minus 6. 1 in 10 to the power of minus 6 means for every 10 lakh plants, spontaneous mutation occurs in 1 plant. 1 in 10 lakhs. Induced mutations. Induced mutations are the mutations we induce by using mutagens. What do you mean by mutagen? The agent which causes mutation can be termed as mutagen. Mutagens are of two types: physical mutagens, chemical mutagens. Physical mutagens. Physical mutagens are again of two types. They are ionizing radiations, non-ionizing radiations. Ionizing radiations, they are alpha rays, beta rays, neutrons. Ionizing radiations are ABN. This is the shortcut. Alpha rays, beta rays, and neutrons. Non-ionizing radiations are X-rays, gamma rays, UV rays. Ionizing means releasing of radicals. Radicals contains free electrons, hence they are very active. UV rays are generally used for the pollen mutations. Chemical mutagens. The chemical mutagens are of four types. They are alkylating agents, base analogues, acridine dyes, and others. Alkylating agents. Alkylating agents usually contribute to mutations by adding methyl group. The most commonly used chemical agent for mutation is the alkylating agent named EMS, ethyl methyl sulfonate. And base analogues. Base analogues are fibromouracil to aminopurine. Acridine dyes. Acridine dyes are riboflavin, proflavin. They are used for frameshift mutations. This is very important bit for any competitive exams. Acridine dyes usually induces frameshift mutations. And others are nitrous acid. It is a deaminating agent. How the mutations works? Mutations increases the recombination rate by reducing the linkage. Linkage is mostly seen where the desired genes are linked with undesirable genes. Mutations by reducing the linkage and increases the recombination frequency releases variability. Dominant mutations frequency is 1 in 1000. Significance of mutations. Mutations provide genetic variability, and it is the only source of variation when the desired genotype is not available in the population, and it also induces cytoplasmic male sterility. And demerits. Demerits are we already discussed that the frequency of spontaneous mutation is 1 in 10 lakhs and dominant mutation is 1 in 1000. Hence a large number of population should be screened even though the mutations are available in the M1 generation itself. Pusa, Tara and Shobha are the varieties of flowers that are obtained by mutations. Shobha is the variety of gladiolus. Let's recall what we had studied in today's Green Coach Horticulture classes. We had studied what is mutation breeding. Mutation breeding is nothing but the induction of artificial mutations for the crop improvement. Hugo de Vares first time coined the mutations. X-rays in plants are used for the creation of mutations by Stadler, and mutations in Drosophila are identified by Muller. Chemical mutations are used by Robson. We had seen what is point mutations. Only occurs in nucleogenes, means only one nucleotide is affected. Gene is the bit of DNA that can be coded for a functional polypeptide chain. Mutations are of two types: the spontaneous and induced mutations. Frequency of spontaneous mutation is 1 in 10 lakhs. Induced mutations are used by using mutagens. Mutagen means the agent which causes mutation. Mutagens are of two types: physical mutagens, chemical mutagens. Physical mutagens are again of two types: are ionizing radiations and non-ionizing radiations. Ionizing radiations are A, B, N, alpha rays, beta rays, and neutrons, and non-ionizing or X-rays, gamma rays, UV rays. Ionizing means it releases radicals. Radicals consist of free electrons, hence they are very active. UV rays are mostly used for the pollen mutations. And chemical mutagens are alkylating agents, base analogues, acridine dyes, and others. Alkylating agents are EMS, MMS, EES. Ethyl methyl sulfonate is mostly used chemical mutagen, and they causes mutations by addition of methyl group. And base analogues, they are 5-bromouracil 2-aminopurine, acridine dyes, riboflavin, proflavin. Acridine dyes causes flame shift mutations. Acridine dyes causes flame shift mutations. This is very important bit. And others are nitrous acid. It is a deaminating agent. Mutation increases the recombination rate by reducing linkage and releases the variability. Dominant mutation frequency is 1 in 1000. Significance: it creates genetic variability and also the cytoplasmic male sterility. DNA screening is very difficult, and the varieties obtained by mutation breeding are Pusa Tara and Shoba. Shoba is a variety of Gladiolus. That's all for now, friends. Thanks for subscribing to Green Coach. If you have any doubts, please comment them in the comment section. We'll assure you that we will clarify your doubts as soon as possible. If you like our video, hit big thumbs up. Please don't forget to like and share. This is Mr. Subbarao along with Hotty Buddy signing off for today. We'll update you soon with another video. Keep supporting us, Team Green Coach.